So we finished with like general overview of the endocrine system. Now let's talk about general properties of hormones. And they can be divided into two large groups based on the chemical structure, based on the properties actually. Hormones can be water soluble. That should probably switch to blue. Blue circle, okay, blue square. And hormones can be lipid soluble, the red one, okay. So water soluble are either proteins, like human growth hormone, or peptides, relatively short chains amino acids, or kind of sort of amine-based hormones themselves, like norepinephrine, have amino group and hydroxyl groups. When the chemical is water soluble, is it polar or not? What do you think? Do you think no? Let's recall the basic properties of water. It's going to be, I'm going to do something, just, come on. Okay. It's not going to be saved, but it's just easier. Okay, so this is just an empty page. Okay, it's not in a lecture, just to draw. Okay, so we have a water molecule. Right? One oxygen, two hydrogens. Okay. All atoms have a certain propensity to pull electrons towards themselves. That's a reminder. It's not going to be on the exam. Remind you about that chemistry. That feature of atoms is called electronegativity. The more electronegative an atom is, the stronger it pulls electrons to itself. Sort of think of it, they crave electrons. Oxygen is second most electronegative atom after fluorine. Okay? So it pulls electrons towards itself very strongly, which means it will pull electrons to itself. Does that make sense? We got it? So electrons, how are they charged? Positive or negative? They're negative, which means when oxygen pulls electrons to itself, oxygen gains partially negative charge. This letter, Greek letter delta, means a little bit. Since the water molecule is neutral, electrically neutral, that little negative charge on oxygen is compensated by, well, technically it's 1.5, right? Partially positive charges on hydrogens. Does that make sense? Okay. So, although the whole molecule is electrically neutral, it is polar. Do you understand what I mean? It has negatively charged end, quote unquote, and positively charged end. Got it? Now, that's the reason why water is liquid. Because partial negative charge of oxygen establishes hydrogen bonds with partially negative charged oxygen establishes hydrogen bonds with partially positive charged hydrogens. That make sense? It's like blah, 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 blah. But picture tells it all, right? Does that make sense? Those hydrogen bonds are pretty strong, okay? They're established by polar molecules. So if you take another polar molecule, say, I don't know, sodium chloride, which is positively charged sodium ion and negatively charged chlorine ion, you put them in water, salt dissolves. Does that make sense? So you take any water-soluble molecule, it must be polar to get dissolved in water. Oil, like cooking oil, does it dissolve in water? No. So it's not polar. Not so, but since it is not polar, it cannot be dissolved in water. Am I clear? So, going back to our hormones. These guys, proteins, amino acids, and they have a lot in common. Proteins are composed of what? Amino acids, yeah. The difference between the peptide and a protein is big, okay? Does that make sense? It's like, like a short chain is a really, really long chain, okay? So they polar. This chemical is polar due to plenty of oxygens and nitrogens that pull electrons to themselves. So all these water-soluble hormones are polar. Let's keep it in mind so far, because that chemical property, water lipid soluble, will determine the fundamental mechanisms of action. Now, lipid-soluble hormones are non-polar, and three main classes that we're interested in are steroid hormones, sex hormones, corticosteroid hormones, glucocorticoids, mineralocorticoids. These guys are lipid-soluble, okay? As are thyroid hormones. So these guys are lipid-soluble because of the iodine. And eicosanoids. Some of this you may have heard about: prostaglandin, prostacyclin, thromboxane. If you've never heard about it, you definitely inhibited the synthesis of those hormones. You know when? When you take ibuprofen, aspirin, or Tylenol. Those are inhibitors of the synthesis of different eicosanoids. These molecules here, we don't really talk about them. Maybe, we should. They are main mediators of inflammation and pain. Say you take aspirin, you inhibit thromboxane. Okay, and that takes away pain and inflammation. Okay, but so far we have fat-soluble and water-soluble. So the pressing question is how these chemical features determine the action of hormones. Well, before we move on to the action of water-soluble hormones, we need to try and recall the very basic idea about the transport of chemicals across the cell membrane. Cell membrane, what is it composed of? So again, no. The membrane, which molecules constitute the cell membrane? Phospholipids. Okay. Now, if we try to picture the very basic structure of phospholipid, it will be the phosphate head, which is phosphate anion and the residue of the glycerol, and two lipid tails. Okay? So phosphate anion. How is it? Is it charged? Anion. Is it charged? Anion. Anion. If you see ion, it is charged and it's negatively charged. So we have negative charge right here. Now, lipid tails are not charged. They're in fact lipid. So you have a charged part, which is, since it's charged, hydrophilic, water soluble, and you have lipid tail, which is not charged, nonpolar, hydrophobic. Does that make sense? So the membrane is organized as the phospholipid bilayer. You have the outer surface of the membrane, well, and the inner surface too, formed by phosphate heads. And inside of it are tails. Sorry, this pen is for some reason not really aligned, so the line is not where I expect it to be, okay? Does that make sense? What it means, on the outside of the membrane, everything is water soluble. Why? You're right, because of the negative charge. What I'm trying to ask is, what is the structural reason why it must be water soluble on the outside? What surrounds the cell? Water. What's inside of the cell? Water, right? Mostly water. So it should be something that can normally interact with water, that can love hydrophilic, love the water. On the inside, it's not. It's like, like you're going to the party. Like, okay, office party. Those are people that you work with. You may despise them deep inside. You may hate them. But you can't just, express it openly. So you put on the mask like you like them. You still, make fun of them in there. But you don't show it. Does that make sense? Same here. The surface of the membrane is hydrophilic. And the inner side is hydrophobic. Now, typical question. Which molecules can cross the membrane? Hydrophilic or hydrophobic? Directly, I mean. Hydrophobic. It's not enough just to get in touch. You have to get all the way through. And only if you can interact with that hydrophobic layer inside, you can get through. Does that make sense? Sometimes compare the cell membrane with a concrete wall covered in pillows. You look from the outside, it's just pillows. You can run through. You try it and you hit the concrete. Okay? If you're hydrophilic, if you like pillows. Okay? Does that make sense? So only hydrophobic molecules can directly, and I want to highlight it, directly, without any assistance, cross the membrane. We talk about gases, respiratory gases, CO2, oxygen, nitrogen, whatever. Small molecules like ethanol can cross the membrane. A lot of lipid-soluble drugs can do that. Okay? But when we talk about water-soluble hormones, can they cross the membrane? They're water-soluble, not lipid-soluble. You have to be hydrophobic, lipid-soluble, to go through. So you have to be hydrophobic, lipid-soluble, to go through. If you hydrophilic, water-soluble, you're stuck on the outside. Does it make sense? That's really, really important. I'm going to ask about this multiple occasions in quiz and in the exam, so please understand that. Are we clear about it? Only hydrophobic hormones can cross the membrane, right? Now, hormones signal, right? So these guys, the hydrophobic ones, they can enter the cell and signal there, no problem, and we'll get to that, but water-soluble cannot get into the cell. So, as Andrew mentioned, they have to interact with a receptor, and since they cannot get into the cell, where is the receptor for them? On the surface, yeah. All hydrophilic hormones, all water-soluble hormones, interact with the receptor on the cell surface. Receptor, receptor, receptor. Okay, those receptors are embedded into the cell membrane. So far, so good. The hormone gets to the receptor, binds to it. Is that clear? Then there are three major options in terms of the hormone. And those three pathways, I'm telling you up front, you must know. Okay? I will tell you to what level of detail you must know those pathways. So pathway number one. They bind to the receptor, so-called G-protein coupled receptor. GPCR stands for G-protein coupled receptor. Okay? They bind to the receptor, and then they trigger the change, conformational change. They activate intracellular G protein. Okay? You follow me? G protein then can do one of two things. Thing number one, it can activate the enzyme called adenylyl or adenylate cyclase. Okay? Adenylate cyclase produces chemical known as CAMP, cyclic AMP. Here it is. And cyclic AMP can do a bunch of things. That's when it comes to an end, what I want you to know. Not like completely, but when cyclic AMP is produced, it can do a bunch of stuff. Am I clear? One of the stuff that it can do, it can lead to activation of the enzyme called protein kinase. I want you to know what kinase does. First of all, the ending of the word ase, A-S-E. What does that mean? Kinase, protease, nuclease. It's enzyme. Yeah, it's generally enzyme, not necessarily breakdown, but it's enzyme, catalyst, okay? So kinase, it phosphorylates the proteins. A lot of new words, I know. Phosphorylation, meaning that you have, let me go up again, just protein plus phosphate gives you protein with a phosphate attached. Kinase facilitates this process. Before we move on, is that understood? Why I pay so much attention to it? This works like a switch. Phosphate is negatively charged. You add phosphate to a protein molecule. Protein molecule changes its shape. Does that make sense? Okay. Have you seen those fancy new car keys? When the actual key can be like folded. Okay. So when you carry it around, you cannot start the car. You need to press the button to release the actual key. Okay. So think about the proteins sort of the same way. Protein is not functional. You add phosphate to it, it changes the shape and becomes functional. Does that make sense? So, cyclic AMP can activate a kinase, which then will phosphorylate the protein. Protein will become active and will do stuff. So, you need to understand the sequence of events. Hormone binds to the receptor. G protein gets activated. G protein activates adenylate cyclase. Adenylate cyclase makes cyclic AMP. And then, all hell breaks loose. Things get activated, here, there. And yeah, know what kinase does. Phosphorylates protein. We're going to run into kinase a couple more occasions. We good? Now another thing that can happen. Go ahead. Say it one more time. There's no specific name. Just it works through cyclic AMP or adenylate cyclase. There's no specific name for the process. It's all a second messenger system. It's all second messenger. You understand what second messenger is? Think about it this way. You come to your friend's house. You knock on the door. It's not your friend who opens the door, but say his brother or his mom. Instead of walking into the house and going to your friend's room, you say, can you please call him? Okay? And maybe like mom says to your friend's younger brother, hey, go get Mike. And then sister, hey, go get Mike. And it's just a chain, okay? You're not getting into the room. Same here. The hormone stays outside of the cell. It leads to the production of the second messenger, which is cyclic AMP in this case. Is that clear? Okay. Another outcome of the activation of G protein is G protein can activate another enzyme called phospholipase. Phospholipase produces chemical called phosphatidylinositol 3. You don't have to memorize that name. IP3. Remember the name of that, the acronym. IP3. It's chemical. Now, this chemical has a very interesting feature. It binds to its own receptors on the smooth endoplasmic reticulum and triggers the release of calcium into the cell. Clear? Now, calcium in this case is going to be your second messenger. It can do a lot of things. It can open channels, close channels, stimulate exocytosis, activate enzymes, bunch of stuff. Again, the point is, hormone interacts with the receptor. That leads to G protein activation. It activates phospholipase C. Phospholipase C produces IP3. IP3 leads to the calcium release. Good? And the third mechanism is probably the simplest. It's mechanism of insulin working. Hormone, right here, binds to the receptor. Receptor has two parts. One part, the one that binds hormone. Another part is the enzyme. It's a kinase. Okay? It directly phosphorylates the protein. Okay, you can see the phosphorylation process. As I mentioned, when protein becomes phosphorylated, this is the phosphorylated protein. It becomes active, things happen. Does it make sense? Go ahead. Huh? Probably. I try to think about analogy. I know. I mean, the idea is that you skip all those steps of, synthesis of this, synthesis of that, synthesis of blah, blah, blah. You have directly the kinase that is attached to the receptor. It's a part of the receptor, actually. Does that make sense, Andrew? Like, imagine that I get stuck in this door, okay? And all you can see is my legs, okay? And my head is out there, okay? So somebody comes to me and starts beating the crap out of me, like hitting me in the face. You don't know what's going on there. All you see is my legs jerking, okay? That's it. So you have the receptor part of the protein that's outside. The effector part is kind of inside. And that effector part modifies downstream proteins that are downstream, activating them, working as a switch. Does that make sense? Before we move on, first of all, it's hard. Didn't say it's going to be easy. It's hard. Let it settle down. I totally expect on Monday questions about it. Okay? I will post a handout where you can the best way to learn it is to picture it. Does that make sense? Try to draw the process step by step in the form of a flow chart, in the form of actual, membrane with all the proteins, up to you. Okay? Does that make sense? It just will help you to process the sequence. We're good. Now let's move on to the lipid-soluble hormones. These guys are a little simpler. Remember, they can cross the membrane, right? So they easily enter the cell, and that's where they interact with their receptor. So inside of the cell, receptor can be in the cytoplasm, okay, or it can be in the nucleus sometimes. Doesn't really matter. Now, I try to focus very much on the concept rather than the details. So I want you to understand that for lipid-soluble hormones, the receptor is inside of the cell. It can be cytoplasm, it can be nucleus. Are we clear about it? Hormone interacts with the receptor, forms a complex. Okay? This complex inevitably ends up in the nucleus. And what is in the nucleus? Which molecule? DNA. What is the function of DNA? What do we need it for? It stores something. Genetic information. Very good. That's it. It stores genetic information. So, hormone receptor complex binds to a specific part of the DNA and initiates the process of transcription, which is the synthesis of RNA that is complementary to a certain DNA fragment. And then when RNA is synthesized, RNA is used as the template for the protein synthesis in the process of translation. Unless you are totally familiar with what I just said and encountered it a million times, you should feel an emptiness in your stomach, really, like you falling from the roof. I came up with what I'm a very humble person, okay? So I think it's absolutely stellar analogy, just brilliant, okay? well, the joke didn't work out that well. My wife thought that it's actually a pretty good analogy of the DNA, RNA, and protein. And I'm going to tell this to you, and you will tell me if it's okay or not. Imagine your DNA is the cookbook, okay? Your family cookbook. Not just the one that you buy in the store, but the one that you keep for generations. It has recipes from your grandma, okay? All kinds of recipes, like 15 recipes for chicken noodle soup, okay? Does that make sense so far? Do you follow me on that? It's huge, gigantic. It has a lot of stuff that is total rubbish, but it's still there. It's your memory. It's your heritage. Of course, you don't keep that kind of family heritage in the kitchen. You keep it in the living room, the nucleus. Okay? Now, you expect guests. This hormone tells you you expect guests. So you want to feed them, right? So you want to cook something. And imagine that your cookbook contains 20,000 recipes. From gluten-free chocolate chip cookies to, I don't know, steak. Everything. Really everything. Are you going to cook 20,000 dishes for your guests? Probably not. Right? You know who's coming. Hormone just told you who's coming. You know which dishes you're going to make for those people. Hormone tells the cell which genes it has to express. Does that make sense? So, as we mentioned, you're not hauling the entire cookbook to the kitchen. In older times, you would probably grab a piece of paper and just scribble down the recipes. Now, you probably just grab your phone, make a picture of the text, bring it to the kitchen, right? And in the kitchen, so that's the picture of the recipe or the scribble of the recipe is the RNA. Is that disposable? Like when you're done cooking, what you gonna do with those pictures? I would probably kill them, just delete them. I don't need them. I have a cookbook, right? Okay, so RNA is disposable, and those pictures go to the kitchen, to the rough endoplasmic reticulum, to the ribosomes, where your cell ultimately makes the meal, the proteins. You cannot feed your guests with the recipe. They don't eat paper. Okay? You need to make an actual meal. It's a protein. Does that make sense? Okay? So, hormone binds to a specific sequence in the DNA. And it will stimulate the transcription of very specific set of genes, which will produce a very specific set of RNA, which will produce very specific set of proteins, which will have very specific effect on the cell. Does it make sense? Okay? Now, this takes a lot of time. So many steps. Really has, takes a lot of time. So, water-soluble hormones, they usually produce very massive and rapid effect. Okay? Because if you think about it, one hormone interacts with one receptor, activates one G protein, activates one molecule of adenylate cyclase, and it starts to churn out cyclic AMPs on and on and on. A lot of cyclic AMPs will activate a lot of proteins downstream, which will activate even more proteins downstream. So it's like an avalanche. Okay? In case of fat soluble, it's very precise, slow, and very, very targeted. So effects are delayed, but they are distributed in time. Longer. Does that make sense? You see the difference? Fat soluble is inside the cell. Before we move on, is that general? You have a general idea of what's going on and what's the difference between water soluble and lipid soluble, right? There's a great likelihood you're going to get your first take-home quiz today. Okay, quizzes are always due next day. I mean, in your case, it's going to be Monday. Okay, and we review them. Don't worry. Okay, I keep scantrons, you keep the quizzes. I don't need these papers. All I need is scantrons to grade. Okay. Now, what can be a stimulus for hormone production? Even without looking on the board, what can trigger the production of a certain hormone? Come on. Stress. Stress, what is that? Is that a change in, is that a nervous experience or chemical changes? Huh? First and foremost, nervous. You perceive, look, anyone afraid of heights? Okay, anyone not afraid of heights? Okay, good. Did you do roller coaster? Did you do roller coaster? Can you do? I would poop my pants if I will go on the roller coaster. Seriously, I'm afraid. This table is as high as I can go without being scared. Okay? So what is stress for me? It's not a stress for you, right? So it's nervous. Nervous experience. It can be a stimulus. Stress. Okay? Again? So, hunger, great, great. Which means change in the nutrient levels. You run low on amino acids and glucose in the blood. Okay? This is called humoral changes. From Greek, humor means fluid, liquid. Okay? Changes in your extracellular fluid. High or low glucose. High or low amino acids. Different changes in your ion concentrations. Whether you just ate a salty meal or you dehydrated. Make sense? It's all humoral changes. Okay? And other hormones. There are several axes. By the way, it's one of the few English words in which singular form and plural form are the same. Okay? Several axes where you have a hormone that stimulates another hormone that stimulates another hormone. Does that make sense? It's pretty common. For instance, growth hormone releasing hormone stimulates growth hormone. Okay? We're clear. So you have to, I will ask you about type of stimulus. Say, I give you an example. When you dehydrated, what type of stimulus it is? You tell me that's humoral because that change in extracellular fluid. You experience stress, nervous. Okay? That makes sense. We're clear. Good. Now, a few basic things. Hormones can act in autocrine, paracrine, and endocrine fashion. Autocrine, acting on the same cell. Okay. Paracrine, acting on the cell that is near. There is no clear criteria when near stops to be near. But, you know, if you have like cells in roughly the same organ, that's paracrine. But most hormones exhibit endocrine signaling. Okay? They release hormones hormones go into the bloodstream and they are delivered to the target cells. Do you understand the difference between these three? Questions? Don't hesitate to ask me questions. Don't hesitate to slow me down. Don't hesitate to interrupt me. Okay? Regulation, I mentioned. Negative feedback. I've already talked about it. High glucose insulin leads to normal glucose, lowering the levels. Okay? Low calcium, release of parathyroid hormone. Your bones get broken down, calcium gets into the bloodstream, calcium back to normal, elevated. It has very important implication in terms of exam. Because if you know stimulus or the effect of the hormone, you can figure out another. Does that make sense what I'm trying to say? So if you remember that parathyroid hormone is produced in response to low calcium, the effect is elevation of blood calcium levels. If you remember that high blood pressure leads to the production of atrial natriuretic peptide, you can figure out the effects. We will talk about, maybe a little bit more steps, but you can think it through. I love endocrine system. If I didn't turn out to be a researcher and would go into the medical field, I would do either endocrine system, endocrinologist, or infectious diseases. Okay, that's very, very logical. Okay, very consequential. We good? Probably the last bit before we get to the actual hormones, how we detect them. One of the unique features of hormones, the vast majority of them, in fact, I know only one hormone that works, that is produced in enormous amounts. It's called adiponectin. All other hormones are produced in the rather, tiny concentrations. So we need to detect them. There are two major approaches to the hormone detection. One is radioimmunoassay, which was developed by the Israeli scientist. I don't remember her name. She got Nobel Prize for it. So before we move on with the detection, what are antibodies? Anyone? Good. Help fight infection. So they're produced in response to something foreign, right? Not only produced in response to foreign, they're very, very specific. Okay? So antibodies that interact with a recognized, say, flu virus will not recognize the common cold virus. They're very different for antibodies. Does that make sense? So what we can do, we can make the antibody that will recognize our hormone of interest. Okay, let's say thyroid hormone. And it will bind it very, very well. Does that make sense so far? Just the whole idea, just what I said. Okay? Now imagine that you have those specific antibodies shown here in gray, okay, that can bind your hormone of interest. And your hormone of interest on this picture, red hormone, it's radioactive. Okay? And you know the exact amount. You already have it. That's your control. Okay? Does that make sense? The sample that you're testing, say somebody's blood, you don't know how much hormone you have in somebody's blood. But you know for sure that the hormone in somebody's blood is not radioactive. Do you follow? So what you do, you mix the known amount of the radioactive hormone with the antibodies, also known amount, and your sample, which is not radioactive. So what's going to happen? Your non-radioactive hormone will compete with radioactive one. Does that make sense? You see what I'm trying to say? So if you have like 10 chairs and you have, I know, no, that's a bad example. Really bad example. Never mind about chairs. So they're going to compete. Okay? So some of the radioactive hormone that would otherwise be bound to the antibodies will become free. Does that make sense? Then what you do, you separate the antibody fraction and the hormones that are not bound to it. Separate them. And then you can measure the percentage of radioactive hormone that is left in the solution or that is bound to antibodies. It doesn't really matter. Does that make sense? Pretty much what happens. The more non-radioactive hormone you add, the less radioactive one will be bound to antibodies. Does it make sense? It is shown here, okay? So you increase the concentration of hormone in the sample, and you see how amount of radioactive antibody that is bound, the amount of radioactive hormone that's bound to antibody declines, because your sample hormone kicks out your control. Does it make sense? Okay, you have stands on the stadium. First Energy Stadium. And initially they have only Browns fans there. Okay? And then you let Steelers fans to get into. And they're gonna, it's a random chance. Either Browns fan or Steelers fan is gonna occupy a particular seat. Does that make sense? The more Steelers fan you're gonna let into the stands, the less Browns fans you're gonna see there. Does that make sense? Do you understand what I'm trying to say? Okay? Same happens here. The more hormone you have in your sample, the less radioactive hormone will be bound to the antibodies. You can plot the calibration curve and then, you can measure it quantitatively. Does that make sense? What I want you to understand about this method is the concept that non-radioactive hormone substitutes radioactive in interaction with antibodies. Okay? Now this method is awfully inconvenient, honestly, because radiation is involved. Much more common method is called ELISA. Not because of a woman. It stands for enzyme-linked immunosorbent assay. We have an antibody again. We have a hormone. So say you don't know how much hormone you have. The more hormone you have, the more complexes like this you're going to have in your sample. Okay? These antibodies, first antibodies, they are immobilized. Okay? So at your sample, you shake it, you wash it, all the hormone that you have gets stuck on the antibodies. Then you add another antibody, and this can be 1, it can be 2. Basically, you have a certain detection system that will change the color, for instance. So the more hormone you have stuck on the plate, the more color change you're going to see. Again, this method is all about detection of a complex between the antibody and the hormone. More hormone, more complexes, higher signal. That's all you need to know. Is that clear? That's a great method. Very easy to do. Very common. Probably the most common method in determining hormones. Okay? Questions before we move on? We have some time to chat about hypothalamus. Unfortunately, I do not have a brain model here. I'm a little hesitant to disassemble this lady. Oh, okay, great. I can pull out her brain. Can everybody sort of see that? That tiny little place right here, the tip of my pen, is hypothalamus. Right under hypo, under the thalamus. Right? That's the chief regulator of your endocrine system. It produces a bunch of regulatory hormones. Okay? I'm very proud of this flow chart. The releasing hormones are green, inhibiting hormones are red. Okay? The table is incomplete. I will tell you why. There is a hormone that was discovered just a few years ago. TRH stands for thyrotropin releasing hormone. Well, guess what? It releases thyrotropin, or thyroid stimulating hormone, from pituitary gland, which must be right here. Okay. CRH, corticotropin releasing hormone, stimulates the release of corticotropin, or another name for this hormone, adrenocorticotropic hormone, from pituitary. Gonadotropin releasing and gonadotropin inhibiting hormone. Gonadotropin releasing hormone releases gonadotropins, follicle stimulating luteinizing hormone. Gonadotropin inhibiting hormone, well, inhibits. This one, it is not in the most textbooks. It was discovered like several years ago. Really, I found the paper 2014 or something. Folks discovered it initially in some animals and then in humans. Okay, so we didn't even know about this hormone. Does that make sense? Growth hormone releasing, growth hormone inhibiting. Acts on the growth hormone. Okay? And finally, prolactin inhibiting factor. Okay? It inhibits prolactin. And guess what? Prolactin regulates milk production. So when this guy is normally produced, females do not produce milk. Does that make sense? Now, a lot of this, the problem with endocrine system, with explaining it, it's very connected. So when we talk about it, my personal reaction would be, okay, so what? No, seriously. I mean, you shouldn't have any idea what thyroid stimulating hormone is, or ACTH, or what are the roles of FSH, follicle stimulating luteinizing hormones. Didn't talk about it. They're parts of different regulatory systems. What I want to highlight here is that these hormones, they stimulate other hormones. That's it. Hypothalamus has sort of a tunnel vision. Hypothalamus works with downstream pituitary gland. Okay, it rarely regulates something else. We'll talk about it. It has some other functions, but it rarely regulates something else. We're good? Okay. On the study guide, I tell you what you need to know, but so far, don't sweat too much over the functions of those hormones, because now it's going to be just a brutal memorization. Unless we get into the business of growth hormone itself, growth hormone inhibiting hormone makes no sense whatsoever. What stimulates it? Why is that produced? Okay. Now, pituitary gland. Quite an enigmatic organ. It's located in stela turcica of the sphenoid bone. You can see hypothalamus right down here. There should be a little stalk called infundibulum, which connects hypothalamus and pituitary gland. Right? It's divided into two very distinctive parts. The anterior pituitary, or adenohypophysis, and posterior pituitary, or neurohypophysis. Okay? Pituitary gland is controlled by hypothalamus. And this stalk, this infundibulum, is very important because it enables that communication between the hypothalamus and the pituitary gland. For a very long time, pituitary gland was thought to be a staple of human hormonal regulation. There were some insane scientists that thought if you would replace pituitary gland of an old person with the pituitary gland of a young person, it will be rejuvenating. Okay? There's actually a very funny Russian novel, very good, by the way. I don't know, there probably should be English translation, about a doctor that replaced pituitary gland of a dog with the pituitary gland of a man, deceased man. And the dog turned into a man. It's hilarious because it kind of has implications of character. It's satire on the society, but the whole idea is pretty funny. Okay, they couldn't live with that dude, what came out of that dog, so they turned him back into the dog. It doesn't work this way. You cannot just replace pituitary gland. It's not going to happen. Okay. Now, pituitary produces a lot of hormones, and these sound more relevant. Growth hormone, obviously, regulates growth. Prolactin regulates milk production. Thyroid stimulating regulates function of the thyroid gland. Adrenocorticotropic hormone regulates the function of adrenal glands, cortisol production. Gonadotropins, follicle stimulating, luteinizing hormone, they regulate the function of testes in males, ovaries, and females. These are produced by anterior pituitary. Okay? They truly produced by the anterior pituitary. These two hormones, oxytocin, which regulates labor, and antidiuretic hormone involved in the water balance regulation, they aren't produced by the pituitary gland. They store it in there. You see the difference? Like, car dealership doesn't make cars. Okay? So, posterior pituitary, or neurohypophysis, it doesn't make anything. It just stores hormones that actually come from hypothalamus. We'll talk about those guys timely. Okay? And then there's a melanocyte-stimulating hormone. We aren't really talking about it. It's technically produced by intermediate zone, but in many cases, it is sort of moved into the group of anterior pituitary hormones. We're not going to mention it. Okay? So, since you're going to have multiple choice questions, you will never have to list all the hormones of pituitary gland. But you have to recognize very clearly, okay? If I ask you, thyroid stimulating hormone is produced by, and you pick pituitary gland. Are we clear about it? Okay, so yeah, I expect you to know that those guys are produced by pituitary gland. Now, the communication between the hypothalamus and anterior pituitary is established in the blood vessels. So here how it works. There is a certain hormone that's produced in the hypothalamus. Let's say it's CRH, okay? Corticotropin releasing hormone. CRH is released into the bloodstream. It's released into the primary plexus, okay? And then from primary plexus through the portal veins, it reaches the secondary plexus. From the secondary plexus, hypothalamic hormone acts on the anterior pituitary gland, which, if it was CRH, will produce ACTH. ACTH enters circulation again, and now is delivered to the entire body. Does that make sense? So you have hypothalamus. Hypothalamus produces the hormone. That hormone enters the hypophyseal or hypothalamic hypophyseal portal system. Portal system refers to those blood vessels. Portal system delivers via blood hypothalamic hormone to the pituitary gland. Pituitary gland responds and releases relevant pituitary hormone again in the blood. Does that make sense? It's all about hormones ending up in the blood. Does that make sense? Good? Now, the anterior pituitary gland produces a lot of tropins. Tropins regulate other hormones. Thyrotropin regulate thyroid hormone. Okay? Thyrotropin is also called thyroid stimulating. Gonadotropins regulate gonads. So follicle stimulating hormone and luteinizing hormone are gonadotropins. They will regulate production of testosterone in males and estrogen and progesterone in females. Corticotropin, or adrenocorticotropic hormone, will regulate the production of cortisol and aldosterone and other corticoid hormones. Okay? So essentially, if you think about this, hypothalamus, again, it's very repetitive, but hypothalamus regulates the production of hormones in the pituitary, which then, those hormones of pituitary gland, regulate hormones downstream in other organs. So you have an axis. Hypothalamus, pituitary, and something else. Does that make sense? So, hypothalamus, pituitary, thyroid. Hypothalamus, pituitary, adrenal. Hypothalamus, pituitary, gonadal. Those are three main hormonal axes in the human body. They're often called HPT, HPA, and HPG axis. And you will see that regulation of those hormones have a lot in common between those axes. Does that make sense? I'm trying to unfold this again because it's so interconnected. Okay? So we're going to come back to the functions of anterior pituitary when we will learn about, say, when we're going to talk about thyroid hormone, we'll have to go back to hypothalamus and pituitary to see how the axis is built. And the same is applicable to the adrenal and gonads. Okay? Does that make sense? Since we're not talking about melanocyte stimulating hormone, yeah, screw it. Okay? It stimulates melanin production, distribution in the skin, so when you get exposed to the sunlight, you get tanned. Okay, and you're protected against sunlight. Got it? We're going to wrap it up now.